Today, we're diving into a scientific breakthrough that could completely rewrite the rulebook on how we fight cancer. We're talking about turning a blunt instrument into a precision weapon. Let's get right into it. All right, let's just kick things off with a huge question. What if medicine was no longer one size fits all? Seriously, imagine treatments designed not for the average person, but designed specifically for you. Well, that's the whole idea behind a field called precision medicine. It's an approach that's trying to move away from that generic model. The goal is to create treatments that are tailored to your unique genetic code, your environment, even your lifestyle. And a lot of people think this is exactly where healthcare is headed. It's a pretty powerful idea, right? And to really see it in action, we're going to look at how scientists are using it to take on one of medicine's oldest and toughest challenges. And that challenge is lung cancer. The stakes here are incredibly high. More than 1 out of every 4 cancer deaths in the US comes from lung cancer. Just let that sink in for a second. That's more than breast, prostate, and colon cancer combined. It's staggering. So what makes it so deadly? Well, a huge reason is that cancer, it's smart. It has this secret weapon, this powerful defense mechanism against our best treatments, and it's called chemoresistance. Okay, so picture this battle. In one corner, you've got standard chemotherapy, these really powerful drugs made to kill off fast-growing cancer cells. But in the other corner, cancer isn't just taking the punches. It fights back. It can actually evolve a defense system to survive the attack, which makes our best treatments totally useless. So you can imagine the mission for scientists was to figure out the source of this defense system, find the linchpin. And they did. They found it. It all came down to a single gene that was basically acting like the cancer cell's personal bodyguard. And that bodyguard has a name, NRF2. The study we're looking at calls it the master regulator of a cell's defenses. So when a cell gets stressed out, say from a chemical attack like chemotherapy, NRF2 just springs into action. It activates hundreds of other genes that build this incredibly powerful protective shield. So let's walk through exactly how this bodyguard does its job. First, the chemotherapy drugs hit the cancer cell. The NRF2 gene senses that stress and flicks on. Then, the cell's entire defense system gets switched on, creating that shield. And the end result? The cancer cell just shrugs off the attack. It survives, and the treatment is a total bust. So this got the researchers thinking, and it led them to this really crucial what-if moment. What if you could take out that bodyguard before the main attack even starts? What if you could just dismantle the cell's defenses ahead of time? And the incredible tool that suddenly makes this idea a reality is CRISPR-Cas9. It's this revolutionary gene editing technology, and people often call it genetic scissors. It basically lets scientists zoom in on one specific gene and with almost surgical precision just snip and disable it. So this opens up a whole new game plan. First, you program the CRISPR tool to hunt down that NRF2 bodyguard gene. Then it goes in, snips it, and disables it. Boom. Bodyguard's gone. The cancer cell's defenses are totally down. And now chemotherapy can finally move in and actually destroy the cell. I mean, it's a brilliant 1-2 punch on paper, right? But the million-dollar question is, would it actually work in practice? To find out, the researchers tested it, first in the lab and then in mice that were given human lung cancer tumors. And wow, the results, they were absolutely stunning. Okay, so look at this. After 16 days, you see that chemo by itself, the tumor just kept growing. The gene edit on its own, it slowed things down a little bit. But look at that last bar. The combination of both, it slammed the brakes on tumor growth, just stopped it in its tracks. The authors used a great word for this. They said the two treatments act synergistically, which is really just a scientific way of saying they're way better together, far more powerful than either one could ever be on its own. And you know, this is so much bigger than just a win against one type of cancer. What this really is, it's a massive proof of concept for that whole idea we started with, precision medicine. It shows that using our deep understanding of genetics to create these smarter, more targeted attacks actually works. Now, of course, we have to be realistic. Getting this from a mouse in a lab to a patient in a hospital, that's a huge leap. And the road ahead is, well, it's complicated. You've got these massive ethical debates. Where's the line between treating a disease and just enhancing a person? Then there are all the regulatory hurdles, huge questions about our data privacy, and maybe most importantly, How do we make sure that when this tech is ready, it's available to everyone, not just people who can afford it?
And all of this leads us to one final, really big question to leave you with. As we get better and better at editing our own genetic code, could this kind of biological hacking really be the key to a future without cancer? It's a pretty wild thing to think about, and this research forces us to take that question seriously. Thanks for digging into this with me.